This week's episode is brought to you by Ecolab. Biotech and pharma leaders, here's an advantage you can count on. Purolite purification resins from Ecolab Life Sciences are engineered for performance, featuring uniquely uniform beads that deliver consistent, high-quality results. With facilities on 2 continents, you can get access to a secure global supply chain and faster time to market. Learn more at puroliteresins.com/ecolab. That's puroliteresins.com/ECOLAB. Welcome back to the Business of Biotech. I'm your host, Ben Comer, Chief Editor at Life Science Leader, and today I'm speaking with Dr. Hernan Bazan, co-founder and CEO of South Rampart Pharma, a company developing a novel non-opioid treatment for pain. Hernan is a practicing vascular surgeon at Ochsner Health and is also the John Ochsner Endowed Professor of Surgery and Innovation. In 2024, he was recognized by the NIH as a Helping to End Addiction Long-Term or HEAL Trailblazer based on the work he's doing at South Rampart Pharma to address the critical need for safer and non-addictive pain treatments in the outpatient and inpatient post-operative settings. Hernan understands the urgent need for safe and effective and non-addictive therapies directly as a surgeon, and I'm excited to learn about the development work South Rampart Pharma is doing, what challenges he's faced as a CEO, his approach to lean company operations, and what's next for the company's lead candidate, SRP-001. Thanks so much for being here, Hernan.
Well, it's really a pleasure, Ben. I was mentioning to you earlier, I've listened to the Business of Biotech now for, I think, over 3 years and enjoy some of the true luminaries you've had, like your CFO, Alan Shaw, and a lot of the zingers that he shares in the practical advice, as well as some of the real innovative founders that are contributing to the biotech and life science field that the United States shares and contributes to the world. So it's a real, true pleasure and honor to be here.
Well, thank you so much for saying that. I have to give credit to my colleague Matt Pillar, who really built the podcast to what it is today. And so I appreciate you sticking with us through the change in host.
Yeah, high quality.
Thank you so much. I want to start as we do on the Business of Biotech with a little about you and your background. You are, as I mentioned in the intro, a surgeon. Why did you decide to become a surgeon initially? What interested you about that field?
It's a good question in terms of, there aren't many surgeons, true, that are doing work in life science or biotech. I realize, obviously, there are many physicians. I'll tell you, when I went to medical school, I had no idea that I would become a surgeon. I mean, there are certainly some people that think of that early on in the clinical years or the 3rd and 4th years of medical school. And that's when you really get exposed to what it is like to be in a clinic. And it was actually my wife, who was a classmate of mine at Georgetown, who did the surgery rotation first and said, you may want to consider this. And it was basically what really solidified it was at the very end of the 3rd year. So I actually was at the NIH in Bethesda, where actually she and I met through the Harvard Hughes Medical Institute as a what's called a research scholar. And it was an incredible 2 years. And so when I finished those 2 years, I thought I was going to go into internal medicine and do some specialty like immunology related or hematology oncology, because I had worked 2 incredible years on immunology and HIV research. And those actual years are important years looking back, because they really influence critical thinking. And so if anyone is listening that's sort of a nascent student investigator and trying to understand between clinical medicine and research and how to marry them, and I know probably we'll touch on the NIH later on. Those types of experiences are real pivotal, because I spent 2 years working in a lab of a real incredible person named Dr. Ed Gardner. He only had 4 people in his lab then. And from that work that he did in 1996, I joined his lab in July. And in May of 1996, he published the first HIV co-receptor. For about a decade, people knew that for the HIV to enter immune cells, in addition to CD4, that there was a second molecule that was needed at the cell surface for the virus to enter, but no one knew what that second molecule was. And Ed, with a group of 4 people, and he was competing with very large labs around the US and a couple around the world, was able to discover the first HIV co-receptor because he was under in building 4 in Bethesda. They're different. The NIH has different building numbers. Building 10 is where I was working when I was my first summer there before I joined his lab. That's the big clinical center. Building 4 is right next to it. That was part of NIAID, the Institute of Allergy and Infectious Diseases. And the head of that at the time was obviously Dr. Tony Fauci.
Right.
And actually, Dr. Fauci had a group of over 100 people, and a significant number of them were looking for the co-receptor as well. As was a big lab at UPenn, a lab in France, a lab at NYU, and one at UCSF, and others. And then with a group of 4, what he did was he utilized the technology that was developed by someone that he was under named Bernie Moss. He was a National Academy of Sciences investigator, and he had cloned all the proteins basically for vaccinia virus. Now, why is that important? Because with that, he was able to do these very efficient expression ability of a way of expressing proteins. And he set up something called a fusion assay. And the fusion assay is what I became fascinated, and that's what he had developed as a way to determine if the HIV envelope protein is on one side and CD4 is on the other, but we are going to basically alternate different molecules on the cell surface to see which one is the most efficient for the fusion assay. And that's how we're going to discover the first. And that's how we elicited fusion, which ended up being a CXC chemokine, CXCR4. And I'll tell you that that paper in Science, I remember before I joined the lab reading it in the New York Times. And then when I joined the lab in July, there was all this activity from all over the world. And a month later, in a 1 week, there were 5 papers in Science, 2 in Cell, 1 in Nature, 1 in PNAS, all about the second co-receptor, CCR5, which is actually the more omnipresent one. And so today, why is all this important? Today, there's actually a therapy, an antiretroviral, maridebruic, that actually targets CCR5 and is part of the antiretroviral. And so, all of these were really important for understanding what's called viral tropism, like which virus will infect which immune cell. Early on, it's CXCR4, but then the envelope protein evolves and then it becomes CCR5 tropic. And it was a pivotal time to understand how to conduct incredible virology, immunology experiments, and then, just to also see the pace of research. My contribution was to demonstrate something called CCR8. It's another chemokine receptor. So that's what I published as I was ending the 2 years, that was important for the infection, HIV infection, the thymus. And, how did this all tie in to the business of biotech? So that was a lot of collaboration with obviously his group in virology immunology where I was working, but also a very good immunologist named Phil Murphy in Building 10, also NID, but a lot of immunology. And from that, the NIH actually patented that and then licensed it. So I didn't know really what was going on. I was just a student, but, bless their heart, they included me in that. The next several years, I would get a $150 check twice a year, which was helpful as a, poor resident,
Absolutely.
month to month. So that was my first sort of assessment of how, really true, important basic science that was translationally targeted, could be potentially commercialized. And now within surgery, then I went to in the clinical years, there actually is a parallel because within surgery, you can make immediate impact on patients. And obviously this is an impact, but it takes a long time. But that has always resonated with me. And then obviously within surgery, there's no more immediate impact in vascular surgery because of the way you can affect patients. And that's a field that has a lot of research and tremendous translational potential as well.
Yeah. Yeah. So let's talk about the circumstances that led you to found South Rampart Pharma. Did that come out of your your experience as a surgeon, or what kind of drove that idea home about, the urgent need for a non-opioid pain therapy?
Right. So This really grew out of a discussion with the other co-founder, the scientific co-founder. It's actually my father, Dr. Nicholas Bazan. He's a fairly well-known neuroscientist. He's got a large group, over 80 people still, with 12 PIs, and they do a lot of work on stroke and neurodegenerative diseases and the neurochemistry of different other things like epilepsy and stroke and Alzheimer's.
Where
Uh,
is his lab?
so they're located actually here in the city of New Orleans at the LA
Okay.
School of Medicine. And they've been very productive and continuously NIH funded since the mid 80s and have um, made seminal contributions in neuroscience and neurochemistry. And he had done something many years ago in pain. So he had a little bit of experience. Many years before that, he had something in ophthalmology in San Francisco with other chairs, and they took the company public. So he had a bit of experience in some contexts. And so we were speaking about obviously the large void in medicine. And there's a colleague in Spain at the University of Alcalá, a medicinal chemist, and what we were thinking was based on the big challenge, which is what was available and still is really available, which is very narrow for pain, and one of them being paracetamol or acetaminophen or Tylenol or Panadol, all the same molecule, and the dose response related issues of liver toxicity. So to address that, what the medicinal chemist did as we were discussing this was make a library of compounds in a rational way, and there's something in the middle of the molecule that prevents the liver toxicity. So since 1983, it's been known that a large amount of Tylenol or Panadol or acetaminophen can cause liver toxicity. In fact, it's still the number one cause of acute pulmonary hepatic failure
Really?
in the U.S. and in many parts of the Western world. And part of the reason has been because many people don't realize it's also present in other formulations, cold and sinus and in other things.
They're
And
taking more than
so
they realize.
people aren't feeling more than they realize. And then, as you know, there's a lot more fatty liver disease, a lot more NASH now. And so there's some susceptibility to that increased dose. And it's a dose response. And so since 1983, when it was published in Journal of Biological Chemistry, NAPKI is a metabolite that causes an injury. And so the way this library was designed was to avoid the NAPKI formation. It's called the benzoquinoamine. So the library that we formed avoids that, maintaining the analgesic or the pain relief. And we were trying to also circumvent the toxicities of NSAIDs, not steroid. I can tell you, I have patients that have had shoulder injuries and they've gone on to dialysis. And obviously, we all know of what the problem is with opioids and abuse potential. And that's what we were trying to do, is come up with a novel non-opioid. Now, even though we were trying to bypass liver toxicity of acetaminophen because these are new chemical entities or NCEs, the regulatory pathway has to be through an RX or a therapeutic, not an over-the-counter. Obviously, it's a different regulatory pathway, which is actually important from a development point of view because obviously the market is larger. And so that's important in terms of attracting partners and so on later on to take this onward. And so that's really how it all grew. And that need, and that was the design. And so then what we did was we were able to secure a large NIH grant. That was the PI from NINDS. They were incredibly supportive. It was what's called a fast track phase 1, phase 2 grant. And that allowed us to do a lot of the characterization. We had already begun doing a lot of the mechanisms of action about how it works in the brain to reduce pain, how it avoids the liver toxicity in finer detail, because of a second mechanism, which is it maintains hepatic tight junctions. And then did a lot of what are called the FDA IND, Investigation New Drug Application Enabling Studies. And a lot of those are, standardized genotoxicity, animal tox studies in 2 species, a lot of metabolite, assessment to make sure they're not toxic and so on, so that you can actually open an IND and get into the clinic. And so that's what we did with that grant. Then the NIH was we were able to secure a second grant, business grant from them. and then raised some diluted funding to get us where we are today, which is now phase two ready, um, having finished a very successful phase one randomized trial.
Yeah, I want to pick up on that in just a second, but I'm curious about your location in New Orleans. Do you have a specific? I mean, your father is based there, his lab is based there. Is there another connection to New Orleans, or a specific reason why you set up shop there?
Well, when we moved from Argentina when I was young, we were younger, and so this is where they had a faculty position. So that's where we were, raised, and I went away to, all my training in different places and for college and then med school and then residency and fellowship. And so actually one of my main mentors from surgery came back to New Orleans from New York, where I did my residency at Mount Sinai Hospital. And so I was his partner for 3 years. And then we stayed here for several years. We've been here for several years. And that's how this grew out. The kids are getting a little older now, and there are other opportunities that are rising. So, the future may evolve. But that's where the company has been headquartered, because a lot of the knowledge has come from there, from the medical school.
Got it. Got it. Well, let's pick back up on SRP-001. You say you're phase two ready at this point. What are kind of the next steps? Are you fundraising at the moment to kind of power that phase two trial? Give me a sense of kind of what's needed, I guess, for the next step.
Sure. So I think anyone listening who's a CEO, co-founder will tell you that every single day you're fundraising.
Yeah, right.
Not a day that you're not. Obviously, there's a lot of strategy and operations and management, but absolutely every day you're trying to evolve the technology. And to do that, you need to secure good capital in an intelligent way. And so we're well on the way with the raise. And so we're aiming to execute the phase two trial as we can finish the race, hopefully within this quarter. So one of the good things about going to meetings is for the last couple of years, I've been able to meet and network with and get to know really well a very experienced individual. His name is Dr. Todd Bertoch. And so Todd is based in Salt Lake City. He's a former anesthesiologist and actually ex-Air Force guy, really reputable and very experienced. And the great thing about him is that he, for now a couple of decades, has taken reformulated well-known drugs that are combined or reformulated and doing acute pain trials. So, and that experience is so critical because pain is subjective. And what one is trying to do in a clinical trial is to objectify something that's subjective, because your threshold of pain may be much higher than mine or vice versa. And so you always have to individualize it to the subject in the trial. And the reason that's important is that you can actually reveal what the endpoints really are, in addition to obviously other things you may be measuring, which is reconfirming safety and pharmacokinetics, how it's absorbed and so on and so forth. And so we've formed that partnership. We've done the protocol, and I feel very confident that the phase two trial will be very successful because of the amount of detail that we've paid to how to enroll, how to measure the primary and then the secondary endpoints in an acute pain situation, and so on and so forth.
Do you anticipate that we will get to a working biomarker for pain? And there's just such a diversity of types of pain that I can't imagine that there would ever be a single biomarker for, pain writ large. But have you seen any studies, or are you aware, of any progress in that direction for specific types of pain that would not be kind of self-reported and subjective, that would be more able to be objectively observed, whether with imaging or whatever else?
Yeah, that's an important question. So actually, in a nutshell, there is no pain biomarker. It's something the NIH actually has been very interested in. And for the previous several years, they've actually had specific RFAs to try to elucidate biomarkers, a pain biomarker. There was a very interesting paper just a few weeks ago in Nature Aging, taking a proteomic approach to look at Alzheimer's biomarkers. And so, for sure, one could take a similar approach of looking, let's say, at acute pain. For example, what our phase trial will be, which is removing of two impacted or partially impacted third molars or wisdom teeth pull, and do an analysis of the blood of the serum and to look for what is a proteomic signature in those subjects compared to, say, placebo, and how are they affected compared to those that receive something like SRP-001, the nominal opiate that we have, versus placebo, or even just control, people that aren't getting any sort of tooth pull, whistling tooth pull. And so, there are, even though none have been revealed to date, perhaps there are approaches like that was just recently done for Alzheimer's that could be applied, for example, in that. But you're absolutely right. Pain is not all the same. So that would be that's more nociceptive pain, acute pain. Neuropathic pain is an even larger problem, clinical problem. Patients that have diabetes, that have diabetic neuropathy, and that may be different than, say, chemotherapy, cisplatin-induced neuropathic pain. That may have its own biomarker. And so, yes, to answer your question, there's no specific biomarker, and likely the different types of pains would have the different biomarker signature once they can be elucidated. But that is an active area of research. So in addition to if a biomarker can be elucidated, that it would be helpful for measuring either primary or secondary endpoints in trials, it would perhaps also be helpful for individualizing treatment on patients.
Right.
In terms of how intense to be and in whom to focus. And we'll talk about this probably at the end, multimodality versus single agent, especially in the future as new pain medicines come on the market that work differently. And then, when then to subject someone to something that has an abuse potential like opioids, which are still going to be the most effective for treating pain, albeit the ones with the highest adverse effect profile. So it could actually not just be just a biomarker for predicting outcomes in trials, but there are many ways that I think it could be helpful clinically. Obviously, it would have to be cost effective. And so right now, it's very much in its nascent form, pre-discovery. But I think one day we'll get there, and those are ways that it could, I think, be utilized.
Yeah, and we will come back to the landscape for pain therapies, but you mentioned cost effective, and that I wanted to follow up on what you were saying about fundraising efforts. You started out the company with some grants, with some diluted funding. You're now raising, I believe, a Series A, and I think, correct me if I'm wrong, targeting an $8 million raise, which is a lot less than a lot of the biotech companies that you see out there raising funds. And I'm curious about, how you might explain that and your kind of approach to lean operations. And does that feed in with this, importance of a new pain therapy being cost effective?
Yeah. So it's, it grabs headlines when you do a 30, 40, 45 million raise for sure. And it's good. Our approach has been different. And the reason that it's been different is because pretty much all the funding that we've raised has gone into the R&D, into technology development and adding value to the venture. And in fact, this was the subject of what you and I were talking about earlier before this, which was the recent Wall Street Journal piece sort of about our lean approach, where, no one is on full salary and it's just fractional work by the people that are working for the company, the team that we formed, which is a stellar team. I'll share, both our CFO, our director of CMC chemistry, our director of toxicology, pharmacology, and our regulatory. And what we're trying to do is move forward as efficiently as possible. Now, if we raised to say 35, 40 million, we could then do a couple of pipelines. We were talking about neuropathic pain, we could do neuropathic pain. And that would take probably about 2.5 to 3 years, many sites. And those endpoints are important endpoints that meet. They're probably not going to be as clear as let's say an acute pain trial, which can actually be executed in well under a year. And this includes writing locking the data lock, writing it, and submission to the FDA. Um, and so what we decided to do, rather than focus on two pipelines, because we were going to do that, as well as we have an intravenous formulation that we developed, an IV and nano suspension. We're going to develop that as well. We decided to, based on some market feedback, on just focusing on acute nociceptive pain, acute pain, to demonstrate, based on how we know it works in the brain and the animal models that we have that demonstrate efficacy, to demonstrate pain relief, to just demonstrate clinical efficacy with a more focused round. And that way it didn't dilute everyone as much, and we could be more efficient in terms of the time frame. And then obviously with this phase 2 data, it would potentially make this a lot more transactable. And so in terms of, moving the technology forward. And so all these things are important, considerations. I think the way we're structured in a lean way is also, a reason that we can forego. And why do that? It's important, right? Because one, obviously, part of the audience may be people that are in operations and so on. You hire someone full time, it's going to be for at least a year at a pretty significant salary. And if he or she is not really producing in 3 or 4 months, you still got to pay them for the full year, and you've lost a year of work. And so we've tried to take a different approach based on context that we have, and so on and so forth. And the sort of team that we formed to use this approach to really just put everything into the R&D and into value formation and forward movement.
Yeah, and I would encourage Business of Biotech listeners to look up the Wall Street Journal piece. When Hernan says that he's running a lean operation, that is not a buzzword. He is paying cash. He does not have full-time salaried employees. He's working on mornings and nights and weekends, and it is actually running a lean organization. One thing that I did that you didn't yet speak to that I wanted to ask about, and I think this is important in the context of opioids as the standard of care for pain therapy, and the fact that they're very cheap, available in generic form. How does this kind of model fit into eventually getting to a price that is ultimately accessible to patients, that is, insurers are going to pay for, et cetera?
Yeah. So that's been one of the big arguments for a while, that what's available is dirt cheap. So why is there a market, or should there be a market? Why put funding into this? And I think this is where Vertex Pharmaceutical should take a lot of credit. So as some may know, some may not, that they've done an incredible job of pushing forward a new set of molecules, and on January 30th, 2025, got FDA approval for acute pain treatment with a molecule that's now called Jornavax, that targets a sodium voltage 1.8 gated channel, and NaV 1.8. Now, why is that important? Is because what they were able to demonstrate about how the market responded to this, it's very positive. So their market cap changed in a very significant way, such that within a week, it was a several billion-dollar upswing in their market cap of the whole company.
So there's a belief that this is going to get used, is what you're saying.
Absolutely. Yeah. Speaking that the market was positive on it. And then when you go to December, when they showed some chronic pain data that we don't need to get into, the market cap also reacted in a certain way. And so these are important contemporary readouts that there is, in fact, a market appetite for it. We all know that clinically there's a huge void. We all know societal there's a huge need void. To the question that you were just asking, though, yes, what's available are generics, and they're cheap. Would people pay for it? So obviously there's a lot of pricing model that goes on, and so on and so forth. And so, from what I understand, their asset is it's a twice-a-day dosing, and it's $31.50 per day. So there can be a premium that can be paid that then will be realized by the investors in a way that it's balanced, so that patients can still benefit. And so it's not a biologic. It's not going to command thousands of dollars, but it's not dirt cheap either. So that I think that the market has demonstrated that, in fact, there is an appetite for paying a premium for something that's new, safe, and effective. Now, regarding their asset, again, they've done a great job with demonstrating this. I think they've said it themselves that this is sort of a first pass,
Right.
and they and others are working on others to improve it. Our approach is a bit different because they're working in the periphery on peripheral nerves. We work centrally in the brain. We know that a radio-labeled SRP-001 crosses the blood-brain barrier. We know where it works in the midbrain, which is called the PAG region, the parental gray area region. And that's what we've published recently. We actually just this morning got notice about a second mechanism in epigenomics and genomics paper that we're revising to be published hopefully very soon as well, to further elucidate the mechanism of action. So we feel very strongly that we know that the mechanism of action should translate clinically to efficacy, because the way SRP-001 works is through production of something called AM404. And that's actually how acetaminophen works. Yet, because it doesn't have the liver toxicity, it's got a much wider therapeutic index. And so what I can share with you, Ben, is that in the hospital, when we gave patients IV Tylenol post knee scope or mini laparotomy, or even a moderate laparotomy or hip replacement, their use of dilaudid or morphine or fentanyl goes down tremendously. These are opioids. And so it's actually at a high dose, it's quite effective. And so that's what we're trying to achieve with SRP-001. IV Tylenol just can't be used for a long time because of liver toxicity, even intravenously. And so we're trying to take a really clinically validated mechanism of action centrally in the brain that, besides AM404, we're elucidating the genomic and epigenomic mechanisms. I think that's important to understand other pain pathways, and are working to demonstrate how this efficacy will be translated with what we'll see clinically.
Right. So this is not targeting the sodium channel in the same way that Vertex's Journavex is.
Correct. And that's important because the future of pain, Ben, will probably be multimodality. So similar to how we treat now hypertension, blood pressure. So, there'll be a calcium channel blocker for blood pressure. There's a thiazide diuretic. There's an ACE inhibitor that may be used in certain patients, a beta blocker. And so there are different classes that can be added to manage hypertension, blood pressure. For chronic pain, there are over 51 million Americans that have chronic pain, the effective treatment of chronic pain, rather than take one asset or combine two that have significant kidney and or liver injury, or put a third that has a significant abuse potential. Hopefully the future will be a couple of assets that work differently, that are effective, and that have a pretty wide therapeutic index that don't have much of an adverse effect profile. And that's how I think we move the needle in the safer treatment of pain.
And potentially personalized to some extent.
Correct.
Yeah. Hernan, you've written that pain is a global juggernaut of suffering, which I found quite poetic, and I wanted to mention it during our conversation, but I'll use that as a lead-in to ask the question. South Rampart was founded in 2016. You're coming up on 10 years as a company. Are you where you kind of hoped you would be starting out in 2016? And what could you say has been the biggest challenge or most surprising challenge that you've faced in this 10 years of work?
Yeah, that's a good question. So I think we're exactly where we envision. We're now a clinical stage with robust human data that demonstrates that SRP is safe, tolerable, efficacy. We've really been able to elucidate robust pharmacokinetics, the PK, again, how it gets rapidly absorbed and then distributed. Now I've optimized the protocol for the phase 2 efficacy. I've established some significant contacts. And so the efficient funding model that we've utilized has allowed us to achieve all these significant milestones. And again, remember, this is from complete synthesis. And so the good news is, we're not taking someone else's intellectual property that we, have to, sub-license or something like that. This is all, fortunately, things that we've developed that have spun out from the university. The founders are in the composition of matter IP that obviously has been nationalized in all key industries. So I think it's where we are coming into just over 8.5, soon to be 9 years. And then I within 12 months we'll be phase 3 ready. And so, as you know, these things, it's never linear. It goes, oscillates up and down, but that's how we view it now going forward. And, regarding you asked what the biggest challenges have been. Again, obviously, anyone will tell you the biotech sector funding is always up and down. So, as I mentioned earlier, constantly in a fundraising mode. NIH has been very supportive. I had a call yesterday early morning. I was in San Francisco at 6 a.m. with my NIH program director, NNDS, who is extremely supportive. And, they're going through some challenges, some significant challenges, facing up to 40% cuts in their. More importantly than that is how the review of new grants has changed now a couple of times just this past few months about how they're reviewed and through which centers and institutes. And so it's unfortunately translated into some reviews. I'll just mention it, just not being quality reviews because of the way they're being handled. And I think they're quickly trying to fix it. I'm very confident in the staff that's there. They're high quality staff, and they're going to get the work done. And there's a lot of very good scientists and leaders within the NIH, and they're going to get this done. They're going to see through this. And so, you just have to be creative when you lead a company like this, looking at, the LUDA from the NIH. ARPA-H is an incredible resource as well. And then being savvy and communicating, not just with investors, financial investors, whether they be VCs or angel groups that have a healthcare assessment, but also engaging early with strategics with pharma and sharing with them what you're doing so you can get feedback and utilize that feedback in how you make decisions in an efficient way.
Yeah. I'm glad you mentioned that you brought up NIH because I think it's not always clear to folks who aren't working directly with the NIH what exactly is happening. It's, if you read headlines, you might think that the NIH is just completely ending all of its funding or an enormous majority of the funding has been halted, not going out the door. People are losing their jobs. But it sounds like you're kind of optimistic about, where the NIH will land as it makes its way through a kind of, period of turmoil, obviously under the Trump administration. But you're somewhat confident in the people that are working there and their capabilities, and B, that their mission will continue. Is that correct, or is there anything?
Oh, absolutely. Yeah, absolutely. I mean, listen, the government has always had the right to prioritize, make their priorities be their priority and emphasize it. That's always been the case. If you look at the history, the last 40, 50 years, they may just change things, a dozen things from year to year. Just this year, there are many things being changed, but that's always in the purview of the U.S. government. The staff at the NIH, the leadership in the institutes, the ones that are the program directors, I can tell you, are very good scientists, very good administrating, and very good at working to do things in an efficient, fair way. I think if all of this helps to increase the efficiency, that's going to be a huge positive. And I think it is becoming a huge positive. There are going to be headlines, but I absolutely have full confidence in the day-to-day operations. And people are going to just adjust and deprioritize certain work and prioritize certain new work to meet the new priorities of the administration. But absolutely, the NIH is going to remain pivotal. And it has to, right? Because, if you look back at 80, 100 years ago, the industry was railroad, oil, and steel. And now the industry is data, biotech, and AI. And listen, we're kind of losing the AI race to China right now. And we may lose it. Hopefully we won't, but we can't lose the biotech and life science to China and EU. And I think everyone understands that's in this field that the way to do that is to keep on supporting the NIH. And obviously then to take technologies like we're doing and many others are doing and then commercialize them. And so I think that adds obviously not only a lot of value to the health and well-being of humans, but also to the economy and jobs and so on and so forth. So I think all those things are going to be realized in a way that it's make the hopefully this ecosystem even more efficient and you just have to adjust.
Yeah, I mean, patience aside, I think it's undeniable that biotech is an economic engine. And in fact, I'm glad you made that point because I know that the EU, we just published an article on Life Science Leader about a number of initiatives that the EU is enacting because they see it as a strategic investment to attract biotech R&D to streamline regulations to get drugs and innovation to the market faster. It's recognized, I guess, is what I'm saying by nearly everyone that there are numerous benefits to having a strong biopharma ecosystem. So I'm glad you mentioned that. We're getting close to the end of our time here, Hernan. I did want to just clarify one thing. SRP-001 is ready to begin phase 2 trials. And I think what you're saying is you're hopeful that at the end of phase 2, you'll have produced some transactable data. Is that ultimately the goal, that you would partner or potentially do a deal with a company to move the asset into phase 3 and ultimately to commercialization, or is it is it possible that you'll pursue that yourself as South Rampart Pharma all the way through to approval? What's your thinking on that?
Right. Good question, Ben. Obviously, there are many options on the table, as you could imagine, when you're at that stage. There's a lot of value in working with a medium or large pharma once it's phase 3 ready with phase 2 data, because obviously the amount, not just the capital, but of resources that they have to do that.
Absolutely.
So we certainly have the context to conduct the phase 3. And then by then, yes, we would have obviously a full-time operations team because it'd be in a different landscape that we'd be operating in to execute those. And then there are the possibilities that certain pipelines are licensed out while others are being developed. So for example, acute and chronic pain may be licensed out while then the company develops the intravenous formulation, because that's its own other application. And/or neuropathic pain has its own, and/or topical. So there are a multitude of possibilities. Obviously, then if there's a significant amount of capital with interest, and then going to public markets is another option by the team at that time that could be also considered. So there are a multitude of opportunities. I think my commitment is to get this developed as efficiently as possible so that once efficacy is demonstrated, safety, it can be utilized to contribute to the well-being of patients.
Yeah, and I should also mention that you have some, I believe, IND-enabling studies kind of in the wings behind this initial acute pain indication with SRP-001. And so that's a potential option for you, is to, bring this lead candidate through, and it's acute pain after a wisdom tooth extraction. Is that right?
Correct. That's the phase 2. That's the phase 2 ready. The IND enabling are more for the intravenous formulation, for the IV, because the phase 2 is for the oral. So there are a multitude of pipelines and possibilities that are in the future.
Yeah, you could potentially, out-license after your phase 2 and use that, turn that back around into R&D on some of your other programs, potentially.
For sure.
Excellent. It's been really a pleasure speaking with you, Hernan. Thank you so much for coming on the show.
No, I enjoyed it, Ben. And listen, again, great job on what you're doing since you've taken this over. I've learned a lot from listening. to many of the sessions that you've had. So great job. Keep it up. And thanks for keeping these discussions out in the open and clear.
Thank you so much. I appreciate that. We've been speaking with Dr. Hernan Bazan, co-founder and CEO of South Rampart Pharma. I'm Ben Comer, and you've just listened to the Business of Biotech. Find us and subscribe anywhere you listen to podcasts, and be sure to check out new weekly video casts of these conversations every Monday under the Business of Biotech tab at lifescienceleader.com. We'll see you next week, and thanks as always for listening.